But what I wanted to kind of talk to you about is the soil health and the soil life and the roles that you can play. Speakers this morning were talking about, sort of, you're the managers in this system, and you play the instrumental role in being able to enhance the life and the biology that's in the soil. And when you think about the soil, I think of the soil as basically, this is the heart of the system. This is the driver. This is where everything happens. And if you think of yourselves as not growing any particular type of crop or raising any type of livestock or anything like that, you're building soil. That's your job. That's what you're going to be doing in this system. And if the soil becomes this thing that is the driver and the thing that has your focus, then you're going to be able to identify tools that you can utilize in order to be able to enhance that. And that's what we're going to be talking about today, is getting those tools and being able to create this system. And there's an incredible amount of value that you can get from the organisms that are in the soil. You can get value in the amount of organic matter that's in the soil. As that's decomposed and processed, that's going to release the nutrients the plant needs to be able to grow. The speaker this morning was talking about a PhD scientist. I'm a PhD scientist. But I'm a PhD scientist. I think that us PhD scientists are learning from people like you and getting that better understanding of what's actually happening out there on the field and on the ground, and being able to recognize the fact that we've got these systems that we can tap into that evolved with the plants. When plants first came on the land, it was a very intimate relationship that happened between the organisms in the soil and those plants and how they were able to grow and produce. And if we can tap into those cycles, that's going to add a tremendous amount of value to our system. So basically what we're trying to do, and this comes from NRCS has a soil health initiative, and what they're trying to do is they're identifying 4 principles that are utilized in order to be able to try and enhance soil health. So the principles are to minimize soil disturbance, to energize with diversity, to keep the soil covered, and to maximize living roots. And that's going to be the things that are going to be driving the biological community. What we want to do, every organism is growing for its own sake, and every organism has the same basic needs. We all wanted lunch, right? The speaker this morning said we all need food. Every organism needs food. We like being inside today as opposed to being outside. We like to have a nice habitat, a nice home, a nice place to live and have shelter. And that's what the organisms need in the soil. And these are the things that we can manage in order to be able to try and get that. So everything that you're doing as you're building soil is, am I feeding those bugs and am I allowing them to keep their home? Those are the questions you have to ask yourself every time you get into the tractor, every time you walk out of your door to go out to the field. Am I feeding the bugs? Am I feeding the soil? And am I allowing them to have a good home? Because that's what's going to be able to provide those economic benefits to you from those organisms that are in the soil. So if we want to minimize soil disturbance, I was in North Dakota for about 11 years with the USDA, and I worked a lot with a lot of really good no-till farmers. Again, I've recently moved to working with the Rodale Institute and working a lot with organic farmers. And there are ways to bridge those ideas and those concepts. There are ways to be able to figure out these tools to minimize the amount of disturbance. The disturbance that you're doing to the soil, that's habitat destruction. This is a wrecking ball. Every time you put an implement into the field, into the soil, that's a wrecking ball going through those homes, those microbial homes. You're destroying those homes. And what ends up happening with that is that those populations, just like, if a wrecking ball came through and destroyed this building and we were having to sit outside today, many of us probably would go home and get sick. And, if it wasn't for some modern technology and things like that, some of us could even die from that. That's what's happening to those microbes, is their populations are declining. The other thing that happens is that resources are then going to be put into rebuilding this building. Instead of resources going and expanding this building and being able to utilize more space and be able to deliver more benefits, resources have to go into this rebuilding process for the habitat that gets destroyed. So every time you do tillage into this, every time you disturb that soil environment, that has to be a rebuilding process. Almost all life on planet Earth is carbon-based. Pretty much everything is carbon-based. So we have to follow the carbon flow in our system, the carbon cycling in our system. All life is carbon-based. Every cell in your body, and every enzyme that you make, all of your proteins, everything is carbon-based. The same is true for the organisms that are in the soil. That carbon comes from the atmosphere in the form of CO2, and it comes into all of this life through the plants. It's the only way that carbon gets into everything is by going through the plants. So the plants have a tremendous amount of work that they need to do. What ends up happening is if it's a rebuilding process, that carbon that could have gone into expanding the populations of the organisms in the soil, that could have gone into the growing of the plant, that could have gone into producing grain, now has to get utilized to rebuild that soil structure, to rebuild that habitat. And that's what's going to affect your bottom line. So you need to think about where the carbon is going and how that's going to be disturbing that system. And the different types of tillage tools that you have can disturb different amounts of the soil. So there are options that are out there. A lot of this stuff, it isn't necessarily a black and white issue. What it is that you got to figure out, okay, how can we have different tools? And again, as one of the speakers said this morning, you guys are the engineers. You're the ag engineers that are out there. You're the ones who have been designing these tools. You're the ones who can figure out what it is. If that is your driver, if my driver is to reduce the amount of disturbance that I have to that soil environment, what is it that I can do to be able to do that? And how can I redesign those tools to be able to reduce that? So we've got some good tools that are going to reduce the amount of disturbance that we have to that soil environment, but we can engineer better ones. We can figure out different ways to still be able to manage a lot of our pest issues, a lot of our weed issues that we utilize tillage for in organic agriculture. We can manage those with some other tools, and then we can maybe re-engineer some of the tools that we currently utilize for tillage in order to be able to reduce the amount of disturbance that we have. How can we do that as a focus? So one of the things we've done at the Rodale Institute is we've engineered this roller crimper. I'm sure many of you have probably heard about it. And basically what this is this is an implement, and it's gone through a few different types of design phases, but this was designed by a farmer at the Institute. The Institute is a research institute, but we have farmers that work there as well. Jeff Moyer is our farm manager, and he's a farmer, and he designed this particular tool. So it wasn't totally PhD scientists that did this. It was farmers with practical knowledge that did this, making this tool. And it was engineered in such a way that it would be able to roll over the field and be able to roll the crops down and create a very good mat that wouldn't have a lot of different, so you wouldn't have a pile of residue here and a more open space here, that it creates a very mono layer. And so, a lot of work went into sort of engineering this tool in such a way that it would roll over the field well, and it has these sort of chevrons on there that allow it to be able to crimp the plant in a number of different places on the stem. And that's going to be harder for then the plant to be able to survive that process. Instead of cutting it in one place or crimping it in one place, it actually crimps it in multiple places on the stem. And that's going to be very effective in being able to kill that plant. So you've got the cover crop that's been in the field. This is a combination of vetch and rye. It's in this field, this particular field, and this was rolled down in the spring prior to planting corn. So this is the corn that went into this mat that was created. And we were just out there this fall. Unfortunately, I don't have pictures of that this fall yet, but we've got very little weeds. There's only a few weeds that actually came up in the row, but other than that, there's very little weeds that came up between the rows when we've got this system. And we were able to retain a lot of this mulch layer in this system. So again, it's an engineering thing. It's a way of designing the tools. Now the other thing that was done was that we put this on the front of the tractor, and then on the back of the tractor we put on a no-till drill. We had some problems because we had this last year, 2 years ago, they had a really big problem because a lot of their seeds weren't getting into the ground very well because they didn't have a very good way of managing the residue and being able to slice into that residue and create a good furrow. So they went out and thought about it and talked with a number of people and talked with no-till farmers and and kind of looked at some of the equipment that was out there and re-engineered some tools to be able to have a better cut of that residue, to be able to cut through that residue better, get a better furrow to be able to put the seed in. So again, it's a much better figuring out. It isn't saying, well, you know, this didn't work, because it didn't work because we had seeds sitting on the ground and we didn't have very good germination, we didn't have a very good stand. So we could have gone this year and said, you know what, this roller crimper thing is junk and we're going to throw it out. But instead, we took a look at what was going on with the system and figured out a way to re-engineer it in such a way so that we didn't have to go back to utilizing tillage as a tool. We could still utilize this as a tool and figure out a way to be able to get in there and plant. And the other thing with this is being able to try and keep the soil covered. And keeping that soil covered with mulch, keeping that soil covered with standing stubble. You don't have to go out there and if you've got, you know, your crop fields where you've got the residue that can stay in the field and be able to protect that soil surface, it's going to protect that soil surface from wind erosion. It's going to protect that soil surface from evaporative demand, from the sun being able to suck water off of that soil surface. So we're able to then protect that soil surface and protect that habitat for the microorganisms that are there by being able to keep that residue on that soil surface and being able to, when we harvest our crop plants, to be able to retain some of the stubble that's there. What are some of the things that we can do? This works very well in a conventional no-till system, but how can we engineer some things, or what are some of the other tools that we can utilize to be able to do this in an organic system? So that we would be able to have good weed management, but still be able to retain that residue and that stubble that's there. These are some of the questions that we're trying to ask and trying to figure out answers for. We want to keep that soil covered because when you hit that surface of the soil with rain, this is from a rainfall simulator, and I hope some of you have been able to see a rainfall simulator in operation. Essentially what happens is we've got these metal pans here. We've got soil samples in there, and there's a sprinkler system that goes over those metal pans, and they're on a slight incline, and you basically can see how much water and soil runs off of that surface. And what ends up happening when you've got even just a gentle rainstorm, they say that a gentle rainstorm on a per acre basis is equivalent to hitting the surface of that soil with about 20 tons of TNT. So this was what it feels like for those microorganisms when you've got a bare soil surface. But if you've got a little bit of protection from the residue that's on the soil surface, you're going to be able to have a system in which that impact of those raindrops gets distributed when it hits that residue as opposed to hitting that soil surface and causing that to break up the structure of the soil, break up those habitats for those organisms, and then that soil is going to run off. When you've got the soil that's covered, you're able to moderate temperatures better. I don't know about you, but when it's 107 degrees, I don't like being outside. I'm not real happy with 87, but that's kind of me. But, you know, it's better than 107. If we're able to keep those temperatures moderated, that's going to allow us to be able to enhance the soil life. They're going to be able to thrive. This is actually at their optimum growth temperature. So you're going to get huge microbial life that's going to be happening at this temperature. You're going to start getting things that are going to start going into a death phase and things that are going to start going into a resting phase until conditions are better. This is in the middle of summer when you'd like to have that biological activity that's happening. If you can keep that soil surface covered, you're able to, again, moderate soil temperatures, but you're also be able to handle weed management better. If you've got this shade that's created here by the plant that you're choosing. The speaker this morning was talking about crabgrass, and basically that he was kind of implying the fact that, well, there's nothing wrong with that, and the fact that he's got cover there. That was the point that he wanted to have. What we can do is we can manage the system so that we can have the weed of our choice, or nature's going to choose it for us. And that's what we need to do, is utilize crops as a tool to be able to manage the system so that we're making the choices, as opposed to allowing nature to make all of those choices for us. So that we can control, we can pick our weed, is essentially what we're trying to do in this system. We're also going to try and get more production in the system as well, energized with diversity. You've got the diversity of the plants that are growing there. This is a cornfield with a bunch of different other plants that have been planted in here. You've got even some things unique as marigolds and cosmos. You've got some clovers in there. So you've got a huge diversity of plants that are growing between the corn rows. We don't need to be able to go out there and keep that area completely clean. Again, we do it because we don't want the weeds there. But I think we underestimate what the plants can do and that the relationships that happen when you have this level of diversity. So there's a lot of things on a microbial level that's occurring here where these plants are actually helping each other out. Oftentimes when we see other plants in our fields that we think of as weeds, we think of that as a problem because it's going to be robbing our crop plant. But what we're finding is that we're recognizing that there are some plants in that system that are actually going to be helping that crop plant. And so if we can figure out which ones to put into that system, we can enhance those benefits. And then we can reduce the amount of weed pressure, the things we don't want to be in the system, we can reduce that pressure and utilize the plants as a tool to do that as opposed to utilizing tillage as a tool to do that. So there are a lot of different options that we have in this system. Utilizing plants with different types of carbon and nitrogen ratios. This is creating more of a balanced diet for the soil environment. Again, those organisms that are in the soil are all hungry. As was said this morning, in about a handful of soil, there are almost more organisms. In a handful of healthy soil, there are more organisms than the number of people who live on planet Earth. So we have billions of organisms in every handful of healthy soil, and they're all hungry. And when you've got this diversity of this diet, you're feeding different organisms. What we want to do is we want to establish an environment in which we've got not just diversity above ground, but we've got diversity below ground. Because those different organisms are going to help us to be able to cycle the nutrients that the plant needs at different times throughout the growing season. NPK is not the whole story, as we well know, when it comes to nutrient management. The microbes that are in the soil are going to feed, you're going to have different populations that are going to feed off of the cereals. This is the residue, the carbon and nitrogen ratio in the residue. Different populations that are going to feed off of the cereals and the corn that are going to be feeding off of the peas that are there. So when you've got those different organisms, that's going to create a balanced diet and it's going to create a healthier soil, just like us. Excuse me. The pie was really good, but if all we ate was pie, we wouldn't be very healthy. We want to be able to have our chicken and our hamburgers and our sausages and all of those types of things as well. That's what the soil needs to be healthy. When you have healthier soil, the other thing that happens again is that it can help to control various pest populations. The greater level of diversity, it makes sure that populations don't grow out of control. A lot of the problems with diseases and pests in our monocultural agricultural systems is the fact they're monocultural agricultural systems. There's no diversity there. We're feeding only one particular type of organism. So no wonder its population is going to get out of control and it's going to cause a problem. It causes a problem because it got overfed, and then its population is such that it's trying to find more food. And so now it causes a disease. It has no predators because we didn't feed anybody else. We can manage these systems in such a way that if we are able to do things to enhance the diversity of those organisms, that's going to provide a lot of these benefits to the system. We also want to be able to maximize living roots. And I think this is one of the biggest things that I've been finding, is having more plants growing in the system, because that's going to feed a wider diversity of organisms. When you've got the living roots that are in the soil, you've got automatically, you have the exudates that come out of the roots, you have the rooting environment itself, you have root cells that as the root is growing through the soil, so you have root cells that are going to get sloughed off, you have leaves that are going to have dropped at different times, you've got the residue and those types of things. You can have decomposers, you can have mutualists, you can have a whole diversity of different types of organisms growing at the same time in the system. So what we want to do is we want to basically manage things in such a way that we're combining some of the different tools, and we're going to get to the point where 2 + 2 no longer equals 4. What ends up happening when we start utilizing the organisms in the soil, it's not like adding one tool to another tool is just going to, build up the system a little bit. There's a huge amount of advantages that can happen with those organisms in the soil. And one of the big things is that they work so closely with the plants that are growing in that environment that they end up doing things in such a way that they'll do things when the plant has those demands. As opposed to us feeding things at certain times and hoping that that's going to be good enough for the plant when the plant needs to go through flowering. I gave it all of that food before, right? So it should be able to do that, and I should be able to get a good yield. That's not what's going to end up happening, because the plant has a different level of demand when it's going through flowering than when it was that you added the fertility to the system. And those organisms are going to be able to provide for those demands. So you've got these organisms in the soil, and they're all kind of connected in this very intricate food web. And I put this up here kind of for a couple of reasons. This is very simplistic design of the soil food web. If we wanted to actually look at all of the different interactions that are happening, you wouldn't be able to see anything on this page. It would be completely black with all the lines that we've drawn, because there's so many different interactions that are happening. When I started school, we talked about the fact that we were able to grow and know about 1% of the organisms that are in the soil. We were able to culture them and have good knowledge about 1% of the organisms that are in the soil. By the time I finished school, people were saying, well, actually, as we've gotten better technology, we understand there's a greater level of diversity that are in the of organisms that are in the soil, that we're now thinking we know about 0.1%. So technology has helped us to realize that we're dumber than we were before. Thank you. It's nice to go to school and realize that I'm really stupid. But basically what that shows is that there's just so many things going on there. It's going to be difficult for us to be able to tap into and identify what all of those organisms are. But what we need to realize is we need to focus on where this food web starts. Because we may not ever understand everything that happens in here. But if we don't have this, none of this happens. And guess where this food web starts? This first trophic level, this initial stage, is the photosynthesizers. It's the plants that are growing there. Now you can add organic matter and you can add residue to the soil, but if you don't have that green stuff fixing carbon, that whole system is going to be limited. So the more that you can have photosynthetic activity, the more that you can have that green stuff fixing carbon, the more that you're going to be able to enhance that system. The more that you're going to be able to provide for this huge, diverse food web. So again, giving you an idea of all of the different types of organisms that are out there in the soil. You have organisms that are associated with plant roots. These are not root nodules that are on there. This is actually a fungus inside the roots called mycorrhizal fungi. You have organisms, you have different types of, this is an algae that's here. So you can have small microscopic plants in the soil, as well as having fungi and bacteria. You have small insects that are in the soil. These are the microarthropods. You have different types of protozoa that are in the soil. So again, you have this huge range of diversity of organisms that come in different shapes and sizes. This organism here, this is a microscopic insect, a microarthropod, but this that's sticking out of here is actually a nematode. That nematode is eating that microscopic insect. This is predator and prey relationships that are happening. So the control mechanism for some of that particular microscopic insect is to have some of these nematodes out there that will kill it, so that its population doesn't get out of control and it starts causing problems. So it doesn't matter if you're a small bacteria, or if you're a large insect, or even a macroscopic insect. you've got small microscopic predators that can prey on you. And that's going to allow for your populations to stay in check and that diversity to happen. This is actually another example of that. This is actually a nematode here. Looks like a worm. And this is a fungus. This is a particular type of fungus. This is very important in some livestock systems because this particular fungus can help to control some of the nematode pests from some livestock, in particular from hogs. And what this fungus does is it forms this lasso or this loop. And it will take the fungal body or just these fine threads. These threads will make a lasso or a loop. And they'll give off, the fungus will give off exudates that will tell the nematode there's food here. And the nematode will come around, and once the nematode puts part of its body through that loop, the fungus causes all of its water inside of its cells to move to the cells that are part of that lasso. So all of the water that was inside these cells here is going to move to where that lasso is. Those cells are going to swell, just like if you have an ankle that you sprain that swells, so the fluid moved to that area. The fluid moves to this area, those cells swell, and that loop closes. And then the fungus gives off exudates that eat that nematode. That's its food source. It's not really, you think you got to sit there and you make a loop and you wait till a nematode comes along and then you get to, but once you caught it, it's a pretty good food source for quite some time. It's a way, again, of being able to control the organisms that are in their soil without us having to add a chemical to kill that nematode. Because the biology is going to do it for us. So again, all of these organisms that are in the soil, they're all starving and they're homeless. And what we need to do is figure out a way to give them food and to keep their house. This whole idea of photosynthetic activity and what happens is we're trying to basically bank carbon into the soil environment. And again, I said that I want you guys to think of yourselves as building soil. You're soil builders. You don't grow a crop. You don't raise any type of livestock, you build soil. Because if you're going to do that, then that's going to do that job for you. You build the soil, it will raise the crop, and it will help you manage your livestock for you. You can basically harness the activity of these billions of organisms that are in the soil, and so this is your money in your system. So everything's got to pay, right? This is all nice and good for me to stand here and say I'm a soil microbiologist. I can tell you to grow bugs, and that's great. Be happy. But this has got to give you some sort of income. Your income in this process is to bank carbon. This is your money that's in the soil. If you can bank carbon into that soil environment, that's what's going to drive those microorganisms. And that's what's going to allow for those nutrients to get cycled when the plant needs them. And that's going to help with the yields. It's going to allow those microorganisms to balance predators so that it's going to reduce the amount of diseases that are going to be happening in the system. It's going to allow those organisms to help to build soil structure, and that soil structure is going to help with keeping that soil on the ground, and it's going to help with water being able to get into that soil environment. So you've got a lot of advantages if you put money in this bank. And to put money in this bank, you just have to have a plant growing. Nothing is exactly free in this system. You got to buy the seed to be able to put that plant in the ground. You have to manage the system in such a way so that plant can grow. But if you do that, you're going to be banking that money into that soil environment. What happens when you bank that soil money into that soil environment is the soil will create part of the habitat that's formed by the soil organisms are these structures called soil aggregates. And they're the pellets that are in the soil. And if you ever look at soil and it's got kind of this cottage cheese type structure, it's got a lot of lumps in it of different shapes and sizes. Those lumps are the habitats for the organisms. They actually they created that. It's a little microbial town that you've got in there. And inside that soil aggregate, you can have bacteria and fungi that are going to be flourishing in there. And it's a very good environment in there because it's all protected. It's like a little neighborhood. It's a little microbial town that's in there. And they've got their food source that's there inside that's all protected inside that habitat. The other thing that happens with having that habitat is that between those clods that are in the soil, between those pellets, those soil aggregates, is open space. You get porosity that's in the soil. And this porosity, you have these openings that are in there, and that porosity is going to allow for when it rains, the water to be able to get into the soil. Reduces, this is the difference between having compacted soils and not having compacted soils, is by having the soil aggregates that are there, having this open space between those aggregates. It also allows for gas exchange to occur. The roots are aerobic, just like we are. They give off CO2. You wouldn't think so, right? Because we always talk about the plants take in CO2. The plants have to respire as well. A lot of their respiration actually comes out of the roots. When a field is flooded, the problem is not that there's water affecting the above ground plant. The problem is the roots can't respire. The roots are being suffocated because the carbon dioxide that the roots give off by respiration can't escape from that root zone. Gases don't dissolve very easily in water. So what ends up happening in the roots in a flooded field is the carbon dioxide that the roots are giving off builds up around that root zone, and it starts to suffocate the plant. It's dying from underneath. Again, the water that's standing on the surface of the field doesn't matter. It's the water that's in the soil. If we can develop a soil environment in which we've got this greater level of porosity, that water's going to go into the soil between rainfalls, and if you've got more porosity there, you're going to have open space in there that those gas molecules can then move into. So that when the roots respire, there's a place for them to go, the gas to go. That CO2 will go through these pores, and it will escape from the surface of the soil. Some of that CO2 can then be absorbed by the plant above ground. It's kind of a cool system. You've got respiration happening below ground. It's the gas that the above ground needs. If you create an environment in which that gas can escape, it'll feed that above ground plant. So you've got this set up there. The organisms that are in the soil, most of them are also aerobic, meaning that they take in oxygen and give off CO2. If we don't have a lot of this open space, the same thing happens with those organisms as well. It creates an anaerobic environment, an environment in which the aerobes can't be able to live and thrive and survive. So you want to be able to have this formation of these soil aggregates. And this is those habitats that are destroyed by tillage. Because those pellets are they're fairly fragile. And when you till that soil, it's again like the wrecking ball coming through this building. This building is our aggregate. That's what the microorganisms have. So these are what the aggregates actually look like. And these are aggregates that have been formed in a relatively short period of time. This is actually in a pot culture. This was 3 months of growth, and you can see all of the little clods that are stuck to the roots. Those are those soil aggregates, all of those little pellets that are stuck to the roots. Some people have said, well, you know what, I've got really sandy soil, I can't make aggregates. I've made aggregates out of pure sand if I'm able to have the roots and the fungi and the microbes that are there that can help to make the aggregates. This is kind of an example of this. This is actually a sandy loam soil initially that was mixed 1-to-1 with pure sand. So you can see all of the sand grains that are in there. It's a huge amount of sand that's there, but again, you've got the roots and you've got the fungi that come off of those roots, and you've got the bacteria that are inside those aggregates that can help to put that together. And that's what you can see here too. This is actually from a farmer's field in North Dakota. Near down, Gabe Brown is going to follow me, and near Gabe, this is actually from Marlon Richter's field, as Gabe probably well knows because I've talked about this a lot, but I think this is a pretty cool one. This is also a sandy loam soil that this farmer has. This was from a cover crop mixture. He had planted a 4-H pea crop. He took that 4-H pea crop off around the 1st of July. He planted this cover crop mixture at July 7th. I collected these roots on August 30th. So in less than 60 days, all of this stuff, these little hairs that are sticking out here, that's all the fungal hyphae coming out of there. These fungi, they're called mycorrhizal fungi. They go into the plant roots and come out into the soil, and they help to form these soil aggregates. And this can happen in a relatively short period of time if you've managed the soil in such a way that there's enough food coming from this plant feeding those microbes so their populations are high enough that they can tap into that when a new plant comes in and be able to form these aggregates very quickly. So this is another way of kind of looking at these aggregates and what we can do with management and how that impacts these aggregates. This is a conventional till spring wheat fallow field, a no-till spring wheat, winter wheat sunflower field, and a moderately grazed pasture. These are all the same soil type. This is from the research farm in North Dakota where I used to work. And so they're all separated by a half mile or less from each other. And what I did was I actually just took the soil and I put it through a series of screens. And I collected, these are called 1 to 2 millimeter aggregates. So it passed through a screen that had a 2 millimeter opening, and it got collected on a screen that had a 1 millimeter opening. So that's the size of these aggregates, are between 1 and 2 millimeters. And all that I did was I put them in these little glass dishes and I added water. And as you can see, the one that came from the conventionally tilled spring wheat fallow system, very few of those aggregates actually stayed together. This WSA is a measurement, a laboratory measurement of aggregate stability. And in the no-till system, more of those aggregates stayed together, but in the moderately grazed pasture, almost all of them did. How do we get here? Because this is what we want to achieve. Because again, making these habitats is an energy-driven process. It's a carbon-driven process. That carbon that's in this system that made those aggregates, between rainfalls, those aggregates get destroyed, and all of the carbon that went into there didn't get banked. Because now all that carbon got released from those aggregates and went back up to the atmosphere. Didn't get banked in the soil. The carbon that went into making these aggregates got banked because it stayed inside those aggregates. This is what we want to do. You made money here, you lost money there, is essentially what's happening in the system. When you don't have good aggregates in the soil, obviously Jamestown, North Dakota, this was in June of 2013, and this was actually near the Jamestown rest area off I-94. And essentially what you've got here is you've got standing water. This is from this area right here, this top picture. And then next to it in the field, you've got a saline issue, salinity issue that's happening here. And the reason this happens is because the water sat here when the snow melted. This farmer had been tilling his field for a number of years. When the snow melted, there wasn't any good aggregate structure because all those habitats got broken up. There wasn't a way for the water to be able to get into the soil. The snow melted, the water stood there until June. Water was still standing there. And it resulted in the fact that the salts that were buried deeper in the soil got wicked up to the surface. Yeah, some soils are high in saline. They have the potential to become saline soils, but they don't have to. How we manage them, the choices that we make, are what causes these things to happen. So this farmer, his solution is to go out here. This is a land roller. His solution is to break up this soil, this salt crust that's on the surface, and break up that soil structure by going over the surface of the soil with a land roller. Because that's really going to help his aggregates. If the tillage destroyed him, think about what a land roller would do to him. Any aggregates that he would have any potential of building here are not going to be able to last that type of thing. The choices that we make in the way that we choose to manage the system can cause us problems or they can solve the problems. We're the ones that are driving this. So, some other pictures. I like to, when I'm going around, and I used to be in this area, so I like to go around and take pictures of things that I see. This is in Montana. These are right across the road from each other. If you were this farmer in this field, you'd say I had really poor soils. It's not my fault. My soils suck. I didn't do anything wrong. I'm limited in what I can do because I have bad soils. What does this guy have? I turned around, literally, on the road. Turned around. So somehow on this side of the road, this guy really got stuck with the bad farm. I don't know how it happened, right? Minnesota, we're losing all of our nice topsoil on our hilltops. It's nice of you guys to do. It's fun because you're going to put it in the ditches. Because then we get to go out there and spend money on cleaning out the ditches, right? So they can raise your taxes so they can go out there and clean the ditches because the ditches are filling with soil, and then they're going to have a problem with the snow filling up and they can't clean the roads very well, and so we got to raise our taxes so they can clean the soil out of your ditches. And the fertilizer that you had in this soil went into the Missouri or the Mississippi and is now in the Gulf. Yay! Way to go. That's fun. This is what we want to do. This is the dirty 30s, or is it? This wasn't going to happen again, right? We've done all of this conservation types of things. We've changed the way that we've managed our system because this wasn't going to happen again. This wasn't going to happen again. Guess what this is? 2012. The way that we choose to manage the system can enhance the microbes that are there and the benefits that they can give us, or we can pay for it ourselves. This is what's costing us money in this system. So you have these organisms that are in the soil, these arbuscular mycorrhizal fungi. I'm going to spend a little bit more time talking about these organisms, one, because these organisms are pretty ubiquitous. They're found in almost all soils all over the planet, and they're associated with pretty much all of the crop plants that we have. And that association is very, very important for nutrient cycling, as well as very important for water management in the system. So you've got these organisms, and they will actually, this is the body of these organisms, are these fine strands. And these strands will actually grow and they penetrate into the root cells. They will penetrate through the root cell wall, and they form these highly branched structures called arbuscules. Highly branched structures called arbuscules. That's kind of neat. We talk about trees and arbor and highly branched. The reason the trees put branches on is because they want to have leaves that can get access to sunlight as the sun passes overhead. That's why they do these highly branched structures, so that they have a greater surface area exposure to the sun. In the soil, you have these highly branched structures that will go out here in the soil beyond the depletion zone around the plant roots and pick up those nutrients, and they form these highly branched structures so they can have a better surface exposure to the soil environment, be more efficient at picking up those nutrients. And then they go inside the plant roots and they form these highly branched structures so they can efficiently drop that stuff off. More surface exposure so that they're able to provide more nutrients to the plant roots. This is the milkman coming to the plant. And the milkman isn't, in town, the milkman came to your front door and dropped this off. For you, that's a very efficient way of being able to get milk. You didn't have to really do much of anything. For the plant, this is a very efficient way for the plant to get nutrients. They're picked up out here, they're shipped by the pipeline inside the roots, and they're dropped off at my front door. What could be better? In this process, right around here, this depletion zone that exists around here, you have the root hairs that are here. The plants will absorb the nutrients that are right around the roots, which is why we talk a lot and we work really hard on being able to make sure that when we've got nutrients, when we add fertility to the soil, when you put organic matter in the soil, you do tillage so that that organic matter, that residue, is buried, and it's going to be buried. You don't till it down real deep in the soil because you want to make sure that it's going to be buried, and that process is going to happen where you're going to be putting the new plant, where you're going to put the new seed. In conventional systems, you have tools that are utilized to make sure, and there's some of them that are utilized in organic systems as well, you make sure that you've got tools that are able to put that nutrient. We do a lot of things with fertilizer placement. We want to make sure that we've got this plate next to the seed, because that's really critically important in the way that we're looking at this. What ends up happening is the reason we do that is because the plants can take up what's right next to their roots, but they can't take up things very easily that are out here. In order to be able to get nutrients that are out here, they have to give off a lot of water to try and get those nutrients to flow back towards the plant roots. This fungus will go out here beyond this depletion zone and pick up those nutrients and bring it into the front door. And it does this all through this cellular pipeline, which means that there's not a lot of water that's used in this process. So you've got, again, this is actually inside the plant roots. What I've done is in the lab, I've been able to take the contents, the plant cellular contents out of there, and it left the cell walls as well as the walls of the fungal hyphae, and then I stained the fungal hyphae blue. So you can see all these highly branched structures that are inside there. And that's showing that that root is very well colonized by this particular type of fungus. This fungus is really good at being able to get especially nitrogen and phosphorus from the soil and deliver that back to the plant. But it can also deliver other micronutrients as well. And it can also help to deliver water. So again, it's got a really important association with how this happens. The other thing with it being able to help with water management is this fungus is really good at being able to form soil aggregates. So I'm going to go back here a second really quick. I said, you had all this fungal hyphae out here that helped to make these soil aggregates. This fungal hyphae, it acts like a net that's out into the soil. And debris will start to collect on that net. It's just like, if you're fishing and you've got, big commercial fishermen, they have these trawling nets that follow behind them, and they get stuff and they always have to clean the nets. This is what's happening in the soil is you have these nets that are out there and debris starts to collect on that. And then the fungus will have that net that's there, debris collects on there, that the fungus gives off exudates that help to keep that stuff glued to that net. So that can help to start forming those soil aggregates. So when you've got those aggregates that are there, the fungus is a big driver in making those aggregates, and as I said, that's really important for porosity. When you've got an increase in porosity by about 45%, you can get an increase in the amount of water that you can hold. The first inch of water the infiltration rate will increase by 167%, and the infiltration rate of the second inch will increase by 650%. This is going to reduce the amount of runoff that you have. That water is going to get into the soil and into the plant roots as opposed to running off the surface. As you increase the amount of this fungus that's there, you're going to improve that porosity, and you're also going to help the plant in managing water. This was a study that was done in the 1950s, and basically it's one of the studies that was used as a foundation for synthetic fertilizer application. And the reason was because this particular scientist found that if he had unfertilized corn, it took 468,000 gallons of water per acre, and it only yielded 18 bushels. But if he had fertilized corn, it took 442,000 gallons of water, and it yielded 79 bushels. So again, synthetic fertilizer, this was kind of the start of the use of synthetic fertilizer, and they used this as an idea for increasing the amount of synthetic fertilizer that we had because fertility mattered. The issue with this and what's happening is, yes, fertility matters, but it matters where you get your fertility from. Because when you add synthetic fertilizer to the system, yes, that can enhance your yield. It can enhance the ability of the plant to manage water better because that fertility that you're adding, there's more of it, and there's more of it around the plant roots. But the plants are going to start sucking up that fertility very early on in the growing season, and then they're going to start running out of it. Because synthetic fertilizers are usually in a chemical formula that doesn't stick around in the soil in that formula. The speaker this morning talked about the fact that the soil has the nutrients that the plant needs, which is very true. The problem is that the plant doesn't know that they're there. The nutrients that are in the soil are in a chemical formula that the plant can't even recognize that they exist. It's essentially like you being shipwrecked on the ocean, surrounded by all the water that you needed, but not a drop to drink. The plant has the fertility that it needs, but not in the form it needs. So synthetic fertilizer came along that said, well, we're going to put the fertilizer in the soil in the form the plant needs, because that's all we need to do. The problem with it is that in that chemical form, it doesn't stay in that soil in that chemical form for very long. Phosphorus gets readily bound up in the soil. When you do soil testing, And again, there was mention about soil testing this morning. A lot of the standard soil testing that's done measures for plant available nutrients, not for total. So they come along and they do a soil test and they say you have very little phosphorus in your soil. It means you had very little plant available phosphorus. You could have a whole bunch of phosphorus in your soil, but it's bound up in a way that the plant can't access. The plant can't access it, but the microbes can do that for the plant. So that fertilizer that's in your soil, that fertility can be tapped into by the biological mechanisms of the soil, by those organisms. And they're going to release those nutrients and put them in a plant available form, and then the fungus can pick that up and ship it through that pipeline to the plant roots. And that's going to be very water efficient, because in this case, where you have the fertilizer that's added synthetically, the plant takes up that fertilizer that's added synthetically, depletes that around the roots, and then it's got to try and get the nutrients that are out here. And its only way of being able to do that is to give off water and exudates to try and get those nutrients to flow back towards those plant roots. It's essentially like saying all of the food is in Minneapolis. There is no more food left around here. So we're going to go out there, and the way we're going to get food from Minneapolis is we're going to put out the interstate. We're going to go up on 35. It's an easy thing, right? Go right up 35, we'll get all the food from Minneapolis. All of the food now, because we're all hungry, all the food's going to be gone from Minneapolis pretty quickly along 35. But there's still a whole lot more food in Minneapolis, it's just out on all of the side roads. So you can go out there and you can try and put more interstates out there in Minneapolis to try and access all of that food. But for the amount of energy that it took to make all of those interstates, you could put a whole bunch of little roads going to all of the little neighborhoods and more efficiently grab all of those resources and be able to take that back to us here in Albert Lea. So yeah, the plant roots are like the interstate. And the fungal hyphae is like all of those little side roads that are out there. It's much more efficient to be able to have all of those little side roads than to have all interstates that are happening. The other thing is if all you had was interstates and you said, okay, I'm sitting here on the interstate and I need to get food from a western suburb that's way far away from the interstate. So the problem is I know where the food is, so what I'm going to do is I'm going to go out there and I'm going to just throw out a whole bunch of resources that way, and I'm going to hope that those resources or that water is going to allow that food to move from that western suburb to the interstate. Some of it will, but not all of it. It's an inefficient way of utilizing water, whereas the fungus can utilize not just the side roads, but it will utilize water more efficiently because it will go from the side roads up to the doors and be able to utilize that as a mechanism, as opposed to just wishing and hoping that the water that you threw out there is going to be able to bring all those resources back. All right, so what has ended up happening in our system is that we've got issues with nutrient use efficiency. And this is what we're trying to make, is a more efficient system. So what we've got here, this graph at the top is actually global cereal yield, which has increased. We've done a lot of things since the 1960s in order to be able to produce more food. The problem is that our efficiency of nitrogen use has actually declined. Now you could look at this graph and you'd say, well, no duh. You increase yield, you got to have more nitrogen. Makes sense, right? This bottom graph is actually the efficiency. It's not the amount of nitrogen that's used. It's the amount of nitrogen that's used per bushel produced. So although our bushels have increased, we've decreased our ability to use that nitrogen well. These old plants here did a better job of utilizing the nitrogen that had given to them than the plants that we have now. So this is what this use efficiency is all about. Our nitrogen use efficiency for synthetic fertilizer is somewhere around 30 to 50%. Our phosphorus use efficiency for synthetic phosphorus is 30% is kind of generous. More of the data is now showing somewhere between 10 and 30%. We're going downhill as opposed to uphill. We've done a lot of things with breeding and genetics to try and get better plants. The problem is that for some reason they're not utilizing our resources any more efficiently. In fact, they're doing it worse. And one of the things that's happened with this is that we've decoupled the needs that come from having the organisms that are in the soil. Many of these organisms that are in the soil, again, this particular fungus by itself is very good at getting nutrients and water from the soil and bringing that back to the plant roots. But it needs to be able to have those nutrients, and it needs to be able to do that in such a way to be able to pay for the carbon it gets from the plant. If the plant has the resources, more than enough fertility early on in the growing season, then that little tiny plant can utilize. That little tiny plant, while it's growing, won't give any carbon up to the fungus because it doesn't need the fungus. Why pay for something that's free, right? Plant's not completely stupid. So it doesn't pay for what's free. The problem is that free stuff doesn't stay around. There was a whole bunch of it when the plant was little, but now the plant needs to go through and it needs to start flowering. And it needs a whole lot of nutrients to be able to do that, because that's what's going to make your yield. It's reproducing now. It takes a lot of energy to reproduce. It needs a lot of food to be able to do that. It needs those nutrients. It didn't work with the fungus throughout most of the growing season, so what ends up happening is the fungal numbers are very small, so the plant now has to try and attract the fungus the best way that it can. And it will give off, in order to attract the fungus, it has to say, I have food here. So it gives off water and it gives off carbon from its roots, telling the fungus that it has food. It's the signal that goes out into the soil environment. But there's very few of these fungi that are around now because they didn't get fed in the beginning of the growing season, so their populations didn't replicate. And so now it has to give off more water and more carbon to try and get these populations growing at a much greater rate. But the plant has a huge nutrient demand. So carbon resources are being stolen away from what could have gone into the grain to try and attract a fungus that could have alleviated the nutrient issues that the plant had throughout the growing season, if the fungus was allowed to colonize those roots early on. So we need to be able to figure out a way to set up this system where we've got these exchanges that can happen within that soil environment. And we need to get the plant working with these microbes early on in the growing season so that before the plant has its greatest needs to be met, it isn't starving and trying to waste a lot of resources trying to get whatever it can from the soil environment. So what we've got is we've got some interesting relationships that we're starting to see too when we've got some of this multi-species diversity that happens. So you've got, this is a grass plant, corn, and you've got a legume here is what that's supposed to be. And essentially in this environment, the roots are all growing next to each other, but the roots can't actually cross between each other. But what we've been able to find is we've actually been able to find that nitrogen that was in this legume can end up in this corn. And the question is how does that happen? Well, the way that that happens is actually through the fungus. The fungus will grow into the roots of the corn, it will grow into the roots of the legume. And it will take nitrogen from the legume and give that to the corn. So in that picture that I showed with all of that diversity of all of those different plants that were growing between the corn rows, you've got nutrients that were actually going from those plants into that corn plant through the fungal hyphae. And this can happen because one of the things that happens here is that nitrogen fixation is an energy-driven process. It takes a lot of energy. For every molecule of nitrogen gas, N2, that's fixed, it can take 32 molecules of what's called ATP. And ATP is basically the energy molecule in a cell. So it's a 32-to-1 ratio. ATP is made up of carbon and phosphorus and a couple of other things, but I won't go into that. But essentially, ATP needs phosphorus in order to be able to have the ATP is kind of the battery. In order for the battery to get charged, it needs an influx of phosphorus. The corn tells the fungus to get excess phosphorus. That's part of what genetically has happened, evolutionary, that's happened between the relationship between the fungus and the corn. So the corn says, get extra phosphorus. That extra phosphorus, the corn will then utilize to pay for nitrogen from the legume. The legume will take some of that phosphorus and it gives it to the bacteria that are fixing nitrogen. That bacteria will fix nitrogen, it gives that nitrogen to the legume, and it gives some of that nitrogen to the grass to pay for the phosphorus that the grass got the fungus to get. And all of this happens through the fungus. The fungus then also has bacteria that grow along the fungal hyphae that are able to release more phosphorus from the soil environment. This is the most efficient mechanism of fertilizer placement in history. Nothing works like this. You couldn't engineer a tool that could do this any better than nature can. And what we've done in the system, the reason that we have reductions in fertilizer use efficiency is we've decoupled this. These organisms are no longer getting fed. And so now it's incumbent upon us to provide those nutrients to the plant instead, which is the reason that we're seeing this decline in nutrient use efficiency. I'm just going to kind of go over really quick. This is from our farming systems trial. This is organic versus conventional tilled systems. I don't know how much time I have left. None? Yeah, okay. I'm good at this. So essentially what we've got is this is the conventional soil. This is the conventional soil, this is the organic soil. Again, conventional organic soil. This is that aggregate stability thing that you can see when we've got that. We're able to build that up because we've enhanced the amount of microbial communities that we've got in the soil sample. One of the other things that we'll see, skip over this, is that we saw a very big impact when we had drought years. So this was a drought year. This corn was planted on the same day. This is the conventional strip here. This is the organic strip. We saw a 30% increase in yield in the drought years. The thing that happens with these soil organisms and working with these biologically based systems is they work really, really well under adverse years, and a lot of times you don't really see much of a difference in average years. And so a lot of people think, well, it didn't work. I put a lot of effort into doing this and it didn't pay me anything. But what it's going to do is it's going to allow the plant to have better resiliency to survive under adverse conditions. And what we've seen, this has been in existence, I'm going to get to the end here, overall comparison. So what we've seen is this particular trial has been in existence for about 34 years. And we've done yield analysis, we've looked at the profitability of the system, we've done energy input analysis and greenhouse gas emission analysis over that time. And what we've found is essentially there's no difference in yield between the conventional and the organic systems. The organic systems are the green bars. The organic systems are more profitable. Even without a price differential, the organic systems can be more profitable because there's a reduced amount of inputs into that system. And then the amount of energy use that goes into the system, even with excessive fertility, or even with excess tillage that can happen in the organic system, it still takes more energy to sustain the conventional system because of all of the inputs and the energy that goes into the creation of those inputs. And then as far as greenhouse gases are concerned, you get more emission of greenhouse gases from a conventional system than from an organic system. So overall what we've seen, and this hasn't happened every year in this way, but overall in about 34 years of history, we've been able to see these differences between these systems. So what I want you guys to do again is to be soil builders. And I think that the new green revolution is a brown revolution. We're going to be cultivating the organisms that are in the soil, and you need to just recognize the fact, and again, every day, tell yourself, am I feeding them and am I keeping them having a home? Whatever it is that you're doing has to do those things. So with that, I just want to thank you, and if you have any questions, if you want any more information, please feel free to contact me.